So now that you have finished watching this lab in action, now that you've gotten to see what the DNA actually looks like in the previous video, I just want to take a moment and make sure that we are filling in the results section for this lab. So I'm on slide number 15. Basically, you are describing what DNA looked like to you. And there is a little picture down here showing you a better example of gathering all of the DNA out of our cup or our beaker. So this right here is another picture of what the DNA looks like. But again, describe what it looks like. So what color is it? What shape is it? Was it wet? Was it dry? Did it remind you of anything else that you've seen before? So that's your job to fill in right here what the DNA actually looked like, which means we can move on to the conclusion section of this lab. And there are some questions to answer over here. So let's answer them together. So the first question is, why did you first squish the strawberries in the plastic bag? And the answer to that, if you don't remember, it was said in the procedure video, but we squished the strawberries in the plastic bag in order to start to break up the strawberry cells. So we had to break down, squished, nope. Why is this so challenging? There we go. We squished up those strawberries because we had to break down the cell membranes of the strawberry cells to release the cell organelles. So then it asks, why did you add the soap and salt to the strawberries? So we added the soap and salt, aka the extraction buffer, to the strawberries in order to help break down the membranes around the cell organelles and expose the strawberry DNA. So basically, we first broke open the strawberry cells, but then because this is a eukaryotic cell, there are cell organelles inside of that cell. We had to break down those cell organelles so that we could get inside of a specific organelle, the nucleus, to get the DNA. So did your results match your hypothesis? Explain. So basically, whatever you predicted the DNA to look like, did the DNA end up looking the way you thought it would? Yes or no, and explain some of the differences or similarities. So you are going to have to fill that question in on your own because you made your own hypothesis. Which part of the lab did you enjoy the most? Again, that is your opinion. So I want to know which part of this lab was the most enjoyable. I know we couldn't really do it in person, but maybe if we had done this in person, what do you think you would have enjoyed? And then which part of this lab did you enjoy the least? So again, maybe if we had done this in person, which part did you not like as much? Yeah, so these three questions, they are your opinion, or it depends on the hypothesis that you made at first, but that would be your conclusion section. We do have one more section to look at really quick. This is the experimental variable section. So this is the control constant, independent variable, and dependent variable. So for this lab, because it was more of an exploratory type of lab, there was no real control for this lab. It just, we can maybe use a picture of what the DNA is supposed to look like, but it is what it is. As far as a constant goes, we kept the strawberries the same. The DNA extraction buffer was the same. There were some sameness between if we had done this in groups. The independent variable could be how long the solution filtered for, because this could affect how much DNA you were able to gather. And then the dependent variable, the thing that we are measuring or recording, or the product we are looking for, is the actual visualization of the DNA. So actually getting that gunk on the popsicle stick, collecting your DNA would be your dependent variable or your result. It's what we were expecting to have happen. So at this point, we have gone through this entire lab. I really hope you enjoyed it. I'm sorry we couldn't do it in person. It is really cool to do this and actually see what the DNA looks like and poke it if you're interested in poking it. But thank you for following along. Of course, let me know if you have any questions or you need help with anything. Make sure that you have answered the questions on slide 17, 15, 9, and 7. And once you do that, make sure you turn this in Google Classroom for full credit. So thank you for watching, and we are done.